So, good evening, guys. Today we'll be starting with the discussion in week 9 of the course in experimental biotechnology. And in this lecture, we'll be discussing about some of the concepts of the microscope and flow cytometry related to the cell biology experiments. Okay, so let's start. So we'll be starting with the first question. Which of the following microscope is better suited for observing thick samples like tissue culture flasks or living cells in culture dishes? The options are upright microscope, inverted, both are equally suitable, or neither of them can be used. Okay, so we have received the first answer. Anybody else?
Sir, inverted microscope.
Okay, so yes, the correct answer is inverted microscope. So the correct answer is option number two, and here is the explanation. So when we are using it, so there are two kinds of microscope: upright and the inverted microscope. Okay. In case of an upright microscope, we have the objective that is focusing upon the specimen that is kept on this particular stage. Okay, but in case of the inverted microscope, the objective lens is just beneath the sample that we are keeping. Okay, so what is the basic advantage of using an inverted microscope in cell culture technique specifically? So what happens, as you might know, that the cell culture plate have this kind of structure, and the cells are basically adhered to the base of the plate. Okay, so in this case, we have the on the cells we will have the media, and while viewing from the top, as in case of an upright microscope, what will happen? The media components will come into the path of the light from the objective to the specimen or the cells that we are keeping. So in that case, what will happen? The path of the light will diverge due to the different refractive index between the plate and the media and the cells that we are viewing. And if, because the cells are on the base, keeping the objective under the cells would be beneficial in this case. That's why we use an upright microscope, because its objective is just beneath the specimen that we are keeping. So we are able to visualize the cells very much clearly as compared to if we would have visualized in an upright microscope. Okay, so let's move on to the next question. What is the primary role of the dichroic mirror in an epifluorescence microscope? To block the emitted light, to allow the excitation light to pass through the specimen, to direct the emitted light towards the detector, or to adjust the magnification. What can be the correct answer? So the correct answer is option number two. And what is the main reason? Here we see. So first we'll see what is an epifluorescence microscope. So in case of an epifluorescence microscope, what is the main thing is the path of light that is falling on the mirror is same as compared to the emitted light. Okay, or the light that is being emitted by the specimen. Okay, so what does that mean? So in that case, the path is almost similar. For example, here in the image, as you can see, that the green light is directly falling on the specimen, and similarly, the path of the light is also taken by the emitted light also. Similar path is taken. So both the paths basically converge or coincide with themselves. That is why it is called as an epifluorescence microscope. Now coming back to the different components, including the dichroic mirror that we are talking about in this particular question. So as you can see that The excitation laser that we are here has a filter that allows only a specific wavelength light as an excited light to fall upon the specimen. And here, as you can see, the incident light and the emitted light follows the same path. So we wanted to use a mirror that only allows certain wavelength of light that is basically falling on the specimen. Okay, but it does not stop the emitted light that is emitted from the specimen. Okay, so that is why it is a dichroic mirror, and it only allows this excitation light uh, to be reflected and fall upon the specimen and not the emitted light through the specimen. Okay, so this is the major reason for the use of the dichroic mirror. And following the application of the emitted light on the particular specimen, then when the specimen emits the light of a specific wavelength, it emits and that falls on a eyepiece or the fluorescence filter followed by the detector. Okay, so this is the main function of the dichroic mirror and the use of the epifluorescence microscope. Okay, so if you guys have any doubt, you can ask. Okay, if not, then we'll move on to the next question. Dash is used for fixing cells for immune localization experiments. The options are 1% Titan X100, BSA, 1x PBS, methanol, and acetone. What can be the correct answer? So here the correct answer is option number 4, methanol and acetone. So this is basically widely used for fixing the cells. Okay, so for example, we have a tissue. So here we are doing it sections. Okay, so why we are doing sections? To basically get a thin layer of cells that shows us the different compartments of this particular organ and the different proteins that are localized at different places in this particular section. Okay, in of this particular organ. Now what we have to do is to detect whether the particular protein is being expressed at that particular position in the organ. We fix the organs first. Okay, so why we fix the organs? We fix the organs such that these the proteins that are present are fixed or localized at the same position. Okay, so this is the major reason why we use fixing, and here we fix in either methanol, acetone, formalin. So these are the common fixing agents that are being used. Okay, and this Triton and BSA, these are used as a blocking agents, and 1x PBS is generally used for washing at different stages of the immune localization experiments. Okay, so let's move on to the next question. Why is fixation of the specimen as important step in immunolocalization experiments? So the options are: it enhances the specificity of primary antibodies, it preserves the cellular structure and prevents degradation of proteins, it amplifies the signal from secondary antibodies, and it is not necessary in immunolocalization experiments. What can be the correct answer? So, as I told you, that if we are using an organ, okay, and you have to visualize different proteins that are there present in this particular organ, then you first fix it. So, what is the major purpose? As I explained in the last question itself, when we are isolating an organ from a living organism, for example, mice that has been usually used as a model organism for various studies. So in that case, what happens? The proteins and RNA start degrading. Okay, so as we are taking out the organs, it starts degrading. Okay, so what will happen? The protein or the protein that we want to visualize and its localization in this particular organ, then that we will not be able to see. So to stop that, we first fix this organ using different agents like methanol, acetone, formalin, etc. So we use these to fix this particular thing, and so that we can visualize the particular protein that is localized in the position which is the its native position in the this particular organ. Okay, so it preserves the cellular structure and prevents the degradation of the proteins. Okay, so this is the correct answer. Okay, so let's move on to the next question. In the immunolocalization protocol, what do primary antibodies specifically bind to? Options are the specimen, the fluorophore, the secondary antibodies, the target antigen or molecule. What can be the correct answer? So, in case of all the immunolocalization protocol, for example, we do immunohistochemistry. So, this is for the tissues. Okay. Then we do immunocytochemistry. This is for basically the cells. Then sometimes we do ELISA. Okay, so these all requires the usage of the primary antibody. Okay, so this term is used to detect the primary antibody basically. And so all the basic principle to use the primary antibody is to detect the specific protein or the protein of interest that we are detecting. Okay, so for example, this is a tissue, and here the protein will be localized here. So we develop monoclonal antibodies basically, or polyclonal antibodies for this particular protein that specifically binds to this particular protein, and it will be further binding to other proteins or the detector that will be used to detect this particular primary antibody. And then we visualize these particular things. So the particular protein that we are visualizing will might be present at the native position in this particular organ. So this is the role of the primary antibody. So the correct answer is option number 4. Okay, so let's move on to the next question. What is the purpose of blocking in an immunolocalization protocol? So the options are to permanently attach the specimen to the slide, to prevent non-specific binding of antibodies, to increase the fluorescence intensity, or to amplify the signal from the primary antibodies. What can be the correct answer? So, as I just told you, we have specific techniques like IHC, ICC, ELISA, or Western blot for the detection of specific proteins. Okay, so what will happen if we are utilizing antibodies for the detection of the specific protein that needs to be detected on, that is under our consideration? So what will happen? This antibody would have a tendency to attach to all the non-specific binding areas. For example, if we are staining a particular cell, and here is the other organs that can be bound. Okay, so in this case, the antibody can non-specifically bind and give false positive results, but our protein of interest would be here. So to stop this non-specific binding, what we do, we block or we cover this surface area, the other regions that can be bound by the proteins by this particular antibody by a blocking agent. Okay, so generally we use 1% Titanex or a detergent. For example, Tween can be used, Tween 20. So these were used, and with the protein like NGS or BSA. So these are basically used. So this is blocking of the particular blocking with the blocking of this particular region, and then we have the that would prevent the non-specific binding of this particular antibody to the other regions rather than to the it will bind only specifically to the protein of interest. Okay, so it prevents non-specific binding of the antibody. This is the major function of the blocking step. So the correct answer is option number 2. Okay, so let's move on to the next question. What would be the maximum resolution that can be achieved if a sample is illuminated by green light, 514 nanometer that is, and observed through an oil immersion objective with a numerical aperture of 1.6? So what can be the correct answer? So the formula for the resolution is lambda by 2 into Na. So lambda here is the wavelength of the light that is being put upon. Numerical Na is the numerical aperture of the objective that is being used to fall the specific wavelength of light on this particular specimen. Okay, so here we have the lambda that is 514 nanometer and 2 into 1.6. So this comes around roughly around 160.56 nanometers. Okay, so here the correct answer would be option number 3. So anything that is related to the resolution. So resolution basically means that how and we can how from how long distance we can detect these two closely spaced points with higher efficiency. Okay, so this is the basic definition of the resolution that we are talking about. Okay, so now, this limit of resolution depends upon the wavelength of light that is being falling upon it, and the numerical aperture of the objective lens that is nearly the surface area that is being covered by the light before falling on the specimen. So this is the specimen, this is the objective. So this particular light waves that is being falling upon the specimen defines the numerical aperture. Okay, so these two components define the resolution by which the specimen can be separated. Okay, so let's move on to the next question. Dash is used to remove water from biological samples from SEM or TEM. The options are washing with gradient solution of ethanol, 5% glutaraldehyde, overnight soak with 1% osmium tetroxide, or air dried in a desiccator. What can be the correct answer? So here two answers can be correct because in case of a SEM or TEM that are electron microscopes. Okay, so electron microscopes, what can be the case because electrons are being used to visualize the particular samples. So they basically are because in case if water would be present, that would hamper the imaging using the electrons. Okay, so in that case we need to dehydrate the samples first, and dehydration is done either by the washing with the gradient solution of ethanol or by overnight soaking with 1% osmium tetroxide. So both of these options are correct. Okay, so 1 and 3 are correct in this case. Okay, so let's move on to the next question. Which of the following is a common conductive coating material used in SEM sample preparation? Options are gold, silver, silicon, or titanium. What can be the correct answer? Okay. So what will happen? Why we use the conductive coating material? So for example, this is the specimen that needs to be visualized for a particular experiment using SEM. And here we have to visualize using the electrons. Okay. So electrons are basically used by conducting the emitting the electrons in a specific manner, so that using the screens the electrons will be imaged and a shadow of the specimen is being constructed. Okay, so naturally the specimens that we have are non-conductive in nature because they are the biological samples. So we have to coat with a conductive material. So here the conductive material that is generally used is gold. Okay, so why gold is used? Because it has high conducting power and it can easily emit the electrons as compared to the other materials. And sometimes titanium is also used, but at that position the cost is also an issue. Okay, so that's why gold is generally used as a conductive coating material in the cases where we need to image the sample using SEM. So the correct answer is option number 1, that is gold. Okay, so let's move on to the next question. What is the purpose of sputter coating in SEM sample preparation? To add the color to the specimen, to make the specimen conductive, to preserve the specimen structure, to remove the excessive fixative from the specimen. What can be the correct answer? So first we'll understand what is a sputter coating. So sputter coating is basically used to coat a thin film of the cell on the thin film of the conductive material on the particular sample that we are used to image the particular sample. Okay, in case of SEM. So now, if we are using sputter coating to coat the particular uh, sample, so in that case, the coating material that we'll be using would be conductive. So in this case, if we are using a conductive material, that means it will make the specimen conductive. Okay. So the correct answer is option number 2. So the sputter coating is basically used to make the specimen conductive. So the correct answer is option number 2. Okay. So let's move on to the next question. In TEM, how do electrons interact with the specimen? The options are: electrons are absorbed by the specimen, electrons bounce off the specimen surface, electrons pass through the specimen forming an image, or electrons scatter randomly inside the specimen. What can be the correct answer? So, there are generally two kinds of electron microscopy that is generally used to study the samples. First is the transmission electron microscopy, and the second is the scan electron microscopy. So this is transmission and this is scanning. Okay, so now first we'll see the case of transmission. Okay, so in case of transmission, as the word suggests, it will transmit through the specimen. Okay, so this is your specimen, and here the electron beam is there. Now it transfers through the specimen, forming an image just kept behind the on the detector that is kept just beneath the specimen. Okay, so it will pass through the specimen and it will form an image. Now, in case of SEM, that is scanning electron microscope, what will happen? We'll make the specimen conductive so that once the electron comes to it, it will bounce back and forms an image. Okay, so now here the detectors would be there that would form a shadow of the particular electrons from the sample and it will show how the specimen looks like. Okay, so it basically gives a 3D image of the sample that we are showing. So in case of TEM, the electrons pass through the sample and in case of SEM, it forms a shadow. Okay, by being emitted from the sample. So the correct answer is option number 3. Electrons pass through the specimen forming an image. Okay, so let's move on to the next question. Which type of cells allow entry of trypan blue to assess viability? What can be the correct answer? The options are only live cells, only dead cells, both live and dead cells, none of the above. Okay, so the correct answer is option number 2, and here is the explanation. So, what happens? Trypan blue is a negatively charged type. Okay, and our cells have a brief negative charge due to the presence of different phospholipids on the cell membrane. Okay, so it is also a negatively charged. So, when the dye comes in contact with the live cells, and live cell shows negative charge, so it will repel. Okay, so it cannot attach to the or enter the viable cells. So this is a viable cell. But in case of dead cells, what will happen? There is a degeneration of the cell membrane of it. Okay, so in that case, what happens? The trypan blue can easily enter the cells and accumulate inside the cytoplasm of the cells. So they are generally permeable to the trypan blue, and trypan blue accumulates inside the cell, just showing that the cells that are blue are being dead or are non-viable. So this is the case of non-viable cells. So trypan blue staining is widely used in case of the cell culture technique, which assess that the cells that we are plating in a plate are viable or not. So what we do, we generally pellet down the cells in a centrifuge and then we treat it with a trypan blue stain and then visualize using a hemocytometer. Okay, so then we'll be telling that the percentage of cells are viable and the percentage of cells are non-viable. So this is routinely used in case of cell culture techniques when we either we have to freeze the cells or have to revive the cells from the freezing medium. So this is the routine culture technique that has been used. Okay, to assess the viability or non-viability of the cells. So let's move on to the next question. In flow cytometry, what is the primary purpose of the forward scatter channel? To measure the cell density, to analyze the cell size, to detect small particles, and to sort cells. What can be the correct answer? So in case of a flow cytometry, what is generally used is, so this is the cell that we are utilizing, and there will be some kind of lasers that is being put upon the cells. So these are able to detect the different properties of the cell, for example size and all those stuff. And here are the detectors that is being applied. So there can be detector here, there can be detector here. So the detectors here are called as the side scatters, that will detect the scattering of the light through the sides of the cell. And here is the forward scatter detector. Okay, so that will help to, if the laser are being put in such a way, then it will able to detect the scattering of the light through these particular lasers in the forward direction. So this is the role of the forward scatter detector. So what it does is, it basically helps in determining the cell size. Okay, so it helps in analyzing the cell size. So the correct answer is option number 2. Okay, so let's move on to the next question. In flow cytometry, the side scatter channel captures data related to the options are refractive index of the cell, cell shape, cell granularity, or cell size. What can be the correct answer? Okay, so just we discuss that this is the cell, and here would be some kind of laser that is being put upon it to detect different properties. And here would be the side scatter channels, here would be the forward scatter channel. So this side scatter channel that we see detects the different uh, properties of the cell related to the scattering of light based upon its different constituents in the cytoplasm. Okay, so now this basically detects the granularity. So what is the purpose of this granularity? Different granulocytes that we study, that have different lobes and different granules. For example, basophils, eosinophils, and many more. So these have different granulated nucleus. So that is why they are called as granulocytes. So these granules are basically detected using the side scatter of the flow cytometer. Okay, so that is why it detects the cell granularity. So the correct option is answer number 3. Okay, so let's move on to the next question. In flow cytometry, what is the purpose of gating? To adjust the laser intensity, to calculate the flow rate of sheath fluid, to select a subset of cells for analysis, or to measure cell size and granularity. What can be the correct answer? So, what happens? There can be a subset of cell. For example, here is the typical scatter plot for the flow cytometry analysis, and here is the population of cell that is being showed by the cell cytometer, and you want only subsequent population of cells that shows a particular property. Okay, so for example, you are differentiating between the viable and non-viable cells, and you see that only these percentage of cells would be only the viable cells. Okay, so now you have to check further properties from these viable cells, so you get this particular population and you use this for further analysis. So the rest of the analysis that will be done will be from this particular population of cells. Okay, so we have to separate the cells based upon their viability or non-viability that been chosen for an example, and the in other cases it can be used for different other properties. Okay, so to separate out subset of cells, we just get the uh, we just put a threshold that is gating for a certain subset of cells. Okay, so this is the correct answer, option number 3. Okay, so let's move on to the next question. Compensation is performed in flow cytometry because of the options are fret between the fluorophores, low intensity of signal, spectral overlap between the two fluorophores, or poor sample preparation. What can be the correct answer? Okay, so what will happen sometimes when we are using two fluorophores? So what generally happens is sometimes, like, this is the emission spectra. So is an overlapping emission spectra for the particular fluorophore or the excitation spectra. Okay, so what will happen if we are choosing any wavelength from here to here, that will allow the fluorescence from these two particular fluorophores, so that will give a mixed result. So to basically separate these, what we do is we compensate. Okay, so to get a sharp peak of the fluorophores from two different fluorophores. Okay, so that this will allow to separate the spectral overlap between the two fluorophores and will give a sharp result between the two fluorophores under consideration. Okay, so this is the basic role of compensation that is being used in the flow cytometry. So the correct answer is option number 3. Okay, so let's move on to the next question. In a cell cycle analysis by flow cytometry, the cells are stained with DNA binding type, membrane specific type, protein binding type, or RNA specific type. What can be the correct answer? So, in case of a cell cycle, if we draw, so the cell cycle is basically the division of nucleus that we study. Okay, so and the different events that takes place in a cell before it goes into the division, and the last stage of the cell cycle is the cytokinesis. Okay, so we divide the cell cycle into basically four phases. So this is the M phase, then we have the G1, S, and the G2 phase. Okay, so before starting with the G1 phase, so it is a preparatory phase. Okay, so cell prepares, grows, and then moves with the increase in the amount of the cytoplasm. The nucleocytoplasmic ratio changes, and the events that moves for the cell division. Then comes the S phase, that is basically the synthesis phase. DNA replication, the major event that happens here. Okay, and the formation of different proteins involved in DNA replication and division. So all these things are formed in the S phase. Followed by the G2 phase, this is the second preparatory phase. So here the rest of the proteins and the RNA and all the steps are being formed. Okay, now comes the M phase, that is the mitotic phase, and it is divided into different phases. For example, prophase, the metaphase, the anaphase, and the telophase, followed by the cytokinesis. Okay, so in the prophase, what will happen? The cell is basically starting to divide, and here the centromeres start to divide with the formation of the astral rays, and the chromatin starts condensing. Okay, so in the late prophase, the chromatin start condensing. In the metaphase, what will happen? The chromatin is condensed into form of the chromatids. Okay, and the chromatids are arranged in the center plate, so that will be easy to divide the particular chromosome. Now in the anaphase, what will happen? The and in this case, what we can see is the spindle fibers are attached with the the kinetochore of the chromosomes. Okay, so now we move to the anaphase. So what will happen here? The chromatids separate from the kinetochore and they start moving towards the different poles. Okay. Now, in case of the telophase, what will happen? They reach the different poles, and now they start to decondense, and here the cytokinesis basically starts. Okay, that leads to the completion of the cytokinesis afterwards. So now, what will happen if we have to study these particular phases using a cell cytometry or flow cytometry? What will we use? Because this is majorly involving the DNA. Okay, so here the major involvement of the DNA binding type would be preferable. Okay, so the correct answer would be option number one, the DNA binding type, for the usage of cell cycle analysis using the flow cytometry. Okay, so let's move on to the next question. In apoptotic cell, annexin 5 binds to phosphatidylserine as the options are: in dead cells, annexin 5 internalize and bind to the PS, that is phosphatidylserine. PS in apoptotic cells translocate to the extracellular side of the plasma membrane. PS moves to the cytoplasm in dead cells and thus accessible to the annexin 5. And PS in live cells move to cytoplasmic side of the plasma membrane. What can be the correct answer? So during a normal cell, we have the cell membrane in an animal cell. Okay, it has two phases. One is the one directs to the extracytoplasmic side, and one directs towards the cytoplasmic side. Okay, so now in these two cases, what will happen? There is a ratio of the phosphatidylserine that is being present. Okay, so majorly the phosphatidylserine is present towards the cytoplasmic side. Okay, so now this is in case of the viable cells. In case of apoptotic cells, what happens? This phosphatidylserine basically shift to the outer side. Okay, so this increases the negative charge on the outer leaflet of the plasma membrane. Okay, so this is the case with the apoptotic cells. So now what happens? The annexin 5 that we are talking about is bound to positively charged cations. So these phosphatidylserine that is being directed towards the extracytoplasmic site binds to this annexin 5. Okay. So this is specifically in case of the apoptotic cells only. Okay. So this is the case with the annexin 5 molecule. So what will happen? Let's see the options. In dead cells, annexin 5 internalized. No, this is not the right option. PS in apoptotic cells translocate to extracellular side of the plasma membrane. Yes, this helps in binding of the annexin 5 to the extracellular side. PS move to the cytoplasm in dead cells. No, this is not the correct option. PS in live cells move to cytoplasmic side. No, this is also correct, not the correct option. So the correct option is option number 2. Okay, so let's move on to the next question. Permeabilization of fixed RBCs on a slide can be done by treating with 1% Titan X100. What is the purpose of this step? The options are to lyse the RBCs, to preserve the internal structure of the cells, to make the membrane porous, or to dehydrate the sample. What can be the correct answer? Okay. So when we are using any detergent, okay, so that could be 1% Titan X100 or any other detergent that we are using, that means that is creating pores in the plasma membrane. Okay, and the pores that is being formed in the plasma membrane permeabilizes the plasma membrane, so that the antibody that we are using to detect the particular protein basically internalize in the cells and will be able to detect the specific proteins. Okay, so this is the basic function of the permeabilization that we are doing. So it breaks off the plasma membrane, so that allows the antibody entry inside the cell. Okay, so it basically helps in making the membrane porous. So the correct answer is option number 3. Okay, so let's move on to the next question. Okay, so this leads to the end of our slide. And if you guys have any questions, then you can ask. Okay, if not, then we'll close the session for today. Thank you guys for joining.